Hey, my name is Marissa Sloan, and today I will start my presentation discussing the isolation of the compound acacetin, and then move on to its inhibitory properties against monoamine oxidase A and B. This paper was originally published in the Journal of Natural Products in 2016 by the authors you see listed here. I'll begin by talking about what exactly monoamine oxidase is, just to make sure we're all on the same page. Then I'll discuss how the researchers were able to isolate the compound acacetin from the plant Calea urticifolia, and how strong an inhibitor of monoamine oxidase it is compared to other known inhibitors. Finally, I'll conclude with some binding characteristics of acacetin with monoamine oxidase as determined by the researchers. Monoamine oxidases are a family of enzymes. MAO-A, shown on the left, is important in the breakdown of monoamines ingested in food, and both isoforms are found in neurons and astrocytes in the brain and spinal cord. They serve to inactivate monoamine neurotransmitters, and because of this, they're involved in the research and treatment of a number of psychiatric and neurological disorders, such as anxiety, depression, Parkinson's, Alzheimer's, some of which can be treated with MAO inhibitors that work to block the action of MAOs. The Calea genus contains over 120 species, most growing in the tropical and subtropical Americas. Some of these species have been used in folk medicine for thousands of years in order to treat things like anxiety, depression, and hypertension. The researchers, in order to discover natural products for the management of neurological disorders, identified plants with inhibitory effects on MAO-A and B. The chloroform extract from Calea urticifolia leaves was found to have inhibitory effects, so the active constituents were examined following a bioassay-guided fractionation. So I'm going to go over the experimental procedure real quick. The leaves were extracted with chloroform and concentrated under vacuum. This extraction showed inhibitory activity of MAO-A and B, and so was fractionated over a silica gel chromatographic column. Fraction F4 showed the highest inhibition, and so was chromatographed by silica gel again, after which F4I was found to be the most active subfraction. This subfraction F4I was chromatographed yet another time to isolate the active compound, and was characterized by proton and carbon-13 NMR. So this is acacetin, the compound isolated and characterized, and it is the very first report of isolation of acacetin from Calea urticifolia. Acacetin is a flavonoid. As mentioned in other presentations, flavonoids are antioxidative, anti-inflammatory, anti-carcinogenic, and so on, as well as able to modulate cellular enzyme function. Acacetin has been used as a dietary supplement and in bodybuilding products due to its anti-aromatase and anti-estrogen properties. It has also been used to suppress neuron degeneration in models of Parkinson's disease. This is a table of the IC50 values of the chloroform extract, fractions, isolated acacetin, and other related flavonoids. IC50 values represent the concentration of a drug that is required for 50% inhibition. So basically, the smaller the IC50 value, the more active an inhibitor the compound is. Acacetin's IC50 values indicate that it prefers to inhibit MAO-B rather than MAO-A by approximately twofold. In comparing acacetin with other structurally related flavonoids, the researchers were able to determine what parts of its chemical structure make it a good inhibitor of both MAO-A and B. So if you look down at the IC50 value for naringenin when inhibiting MAO-A, it is many magnitudes greater than that of acacetin, making it a much poorer inhibitor. The IC50 value for quercetin is also significantly larger than that of acacetin. The researchers were able to determine that hydroxyl groups at positions 5 and 7 in ring B and a double bond between positions 2 and 3 in ring A are crucial for MAO-A inhibition. The absence of this double bond, as you can see in naringenin on the left, reduces activity. At the same time, if there's more than one hydroxyl group at ring C, as seen in quercetin on the right, activity is reduced as well. But that's just comparing inhibition of MAO-A. MAO-B is a similar but structurally different enzyme, and so compounds will have different inhibitory activities for it. As you can see, acacetin's IC50 value for MAO-B is much smaller than those of apigenin and galangin, indicating that it is a more active inhibitor. In fact, there's 75 times more MAO-B inhibition by acacetin than galangin. The researchers were able to determine that an oxygenated 4' position at ring C is crucial for MAO-B inhibition, as can be seen to be absent in galangin. Additionally, a methyl group attached to the oxygen at this position, as opposed to just an OH group in apigenin, also increases activity. Then the researchers wanted to determine whether MAO-A and B inhibition by acacetin was competitive, non-competitive, or mixed. In competitive inhibition, an inhibitor molecule is similar enough to a substrate that it can bind to the enzyme's active site to stop it from binding to the substrate. The researchers tested acacetin against MAO-A at different concentrations of the non-selective substrate kynurenine, as indicated by the x-axis here. The control has no acacetin, one concentration of acacetin is below the IC50 value, and the other concentration is above the IC50 value. As you can see, the y-intercept on the Lineweaver-Burke plot is the same whether the concentration of acacetin is 0, 0.125, or 0.25 micromolar. And these results suggest MAO-A inhibition by acacetin is competitive. The researchers did the same for MAO-B, testing acacetin against MAO-B at different concentrations of the non-selective substrate kynurenine. The results suggest MAO-B inhibition by acacetin is also competitive, as indicated by the consistency of the y-intercept despite changes in acacetin concentration. The researchers also created Lineweaver-Burke plots for the inhibition of MAO-A and B by phenelzine, which is a standard MAO inhibitor, in order to compare their Ki values. Ki values indicate how potent an inhibitor is. They're the concentration required to produce half-maximum inhibition. So basically, the smaller the Ki is, the more potent an inhibitor the compound is. As you can see in the table, acacetin had similar Ki values for MAO-A and B, despite MAO-B having an IC50 value that was under half that of MAO-A, if you remember. Acacetin also had much smaller Ki values for both MAO-A and B than those of phenelzine, which means it's a stronger inhibitor. In order to test whether this inhibition was time-dependent or independent, the enzymes were incubated with either phenelzine or acacetin for 15 minutes. The control on the top line didn't contain inhibitors. The results show that inhibition of MAO-A by acacetin wasn't time-dependent, as you can see by the steady activity for 15 minutes, compared to phenelzine, which dropped significantly from minutes 1 to 5. I won't go into the same amount of detail here, but the exact same test was done with MAO-B, and the results showed that inhibition of MAO-B by acacetin was also time-independent. Dialysis was used to measure whether or not the inhibition was reversible. A high concentration of inhibitor was incubated with MAO-A and B for 20 minutes, and then the mixtures were dialyzed overnight. The activities were analyzed before and after that dialysis, and the results show that after incubation, acacetin inhibited over 95% of MAO-A activity, but over 90% was recovered after dialysis. After incubation with MAO-B, 90% of enzyme activity was inhibited but was fully recovered after dialysis. This suggests MAO inhibition by acacetin is reversible due to the dissociable nature of their enzyme inhibitor complexes. A docking simulation was used to investigate the binding modes of acacetin with MAO-A and B. The active site of MAO-A, as shown in pink, contains 6 water molecules, shown in red, that form at least 2 hydrogen bonds with non-waters. MAO-B, shown here in green, has 10 water molecules in its active site, and these are shown in gray. TYR326 in MAO-B imparts more polarity to the active site, attracts waters, and forms strong hydrogen bonds. Its counterpart in MAO-A, ILE335, is hydrophobic. Water molecules W1 through W5 are in the same area of both enzymes, but W6B through W10B are in a different area of MAO-B and completely absent in MAO-A. Docking is a modeling method which predicts the preferred orientation of a molecule to an enzyme, and the docking score is the predicted binding affinity. Acacetin binds more favorably to MAO-B with a better docking score of negative 8.1 kilocals per mole than MAO-A's docking score of negative 6.2 kilocals per mole. This confirms the researchers' theory that acacetin more tightly binds to MAOB by forming a network of hydrogen bonds. So in conclusion, Calea articifolia is a source of the flavonoid acacetin, and this research was the first time it had been isolated from the plant. Flavonoids have attracted attention for preventing neurological disorders, and so acacetin was tested for its inhibition of MAO-A and B. It was found that it reversibly and competitively inhibits both MAO-A and B, independent of time. Although it binds more favorably to MAO-B by forming a network of hydrogen bonds with surrounding water molecules. And that concludes my talk. Thank you and stay safe.